Now we can study another maternal inheritance, that is cytoplasmic male sterility. So what is cytoplasmic male sterility? Actually, this is a condition that can be commonly observed in some of the higher plants. In these higher plants, they are unable to produce a functional pollen. So these kinds of conditions means in this higher plant they are unable to produce a functional pollen. These conditions can be used as a valuable tool for the production of hybrid plants in some of the self-pollinating crops like cotton, rice, maize, and some other vegetable crops, etc. This cytoplasmic male sterility that has been identified in more than 150 different plant species. In the case of higher plants, we can observe female sterility and male sterility. The male sterility that is more prevalent than that of the female sterility. You know why? There are reasons. Because the male sporophyte and gametophyte simply pollens. They are less protected in the environment than that of the ovule or embryo sac. You all know well that the pollens are less protected in the environment. They may be carried across by means of wind or some of the pollinators like bats, bees, and some of the birds, etc. But the ovule and embryo sacs, they cannot be carried in that sense through the environment. So these ovule or embryo sacs are protected in the environment. So here, what is the advantage of that? Because the mitochondrial genes which are maternally inherited, they are still protected in the ovule or embryo sacs. So there is no need to be worried lot much about the pollen production. The male sterility that can be assayed through some of the staining techniques like carmine, lactophenol, iodine, etc. So this is a picture of different kinds of pollens. They were stained with some of these techniques. Here you can see some of the normal pollens, and see these are some of the abnormal pollens. So by means of these sorts of staining techniques, you can distinguish a normal pollen with that of the abnormal one, or that bears a cytoplasmic male sterile one. But for the female sterility, that can be detected by the absence of seeds in that plant. That means we can propagate the male sterile plants, but we cannot propagate a female sterile plant because they cannot produce a proper seed at all. The male sterility that can be generated by a mutations in the nuclear or cytoplasmic genes. That means they may be cytoplasmic or cytoplasmic genetic one. The CMS technology that relies on three kinds of plant lines. The first one is male sterile lines, second one is connate maintainer lines, and the third one is restorer lines.